Okay, so I'm going to be at the other end of the spectrum. Just to give you a little bit more of an introduction to myself, I'm a neonatologist, and I work in a neonatal intensive care unit at the University of Florida. We have 72 beds. The kind of patients I take care of are largely premature babies down to now 22 weeks gestational age. I think that age has really decreased markedly over the last couple of decades. When I first started in neonatology, we were caring for babies occasionally less than 28 weeks gestation. Now we're down to 22. So this is a remarkable challenge, especially when it comes to nutrition of these infants. The talk I'm going to give is largely going to be a general talk, and I'm not a veterinarian, and I don't know very much about specifics of companion animals, but I think that some of the aspects of the developing gastrointestinal tract that I'm going to be talking about, and some of the immunonutrients, especially in terms of what we find in mother's milk, is going to be important for this lecture. So I'm going to talk about the intestine as an immune organ, the innate immune cells and functions, interactions of the intestinal barrier with the microbiota and linked to adaptive immunity, and then some clinical correlations. The intestine is frequently thought of as primarily a digestive, absorptive organ, but it's become very clear that it's much, much more than that. It's the largest immune organ of the body, and certainly represents a major organ that plays a role in many, many diseases. When I first started working in the area of mucosal immunology, one of my mentors told me that the gut is the motor that drives systemic inflammation. And I began to realize how important that concept was, because many of the disease processes that we see in our babies are not just in the gut. We see them in the lung, in the brain, various other organs of the body that start with the gastrointestinal tract. The gut also harbors a huge microbial ecosystem, a couple of kilos of microbes that are especially found in the distal gastrointestinal tract, and I'll talk about that. And it also harbors the enteric nervous system, and if you put all the nerve cells from the gastrointestinal tract together, it equates to that of our spinal cord. So tremendous nervous system in our gastrointestinal tract. The surface area of an adult human equates to that of a tennis court, 200 square meters. And this is made up by the villi and the microvillous structure of the intestinal epithelium. And when we think about the intestinal surface, there's one layer of epithelial cells that separates the intestinal lumen from a highly immunoreactive subepithelium. And breakdown of this single layer of epithelial cells can have tremendous consequences. The gut content of microbes is huge. It's 10 to the 13th cells. An individual consumes 2,500 kilograms of food antigen during that individual's lifetime. And this single layer of epithelial cells is the difference between those antigens and a highly immunoreactive response. We have two components of the immune system: the innate immune system, which is more primitive, and this is barrier function, various other components that act immediately, versus adaptive immunity, which is the component of the immune system that has to be educated. Vaccines are an example of how we stimulate the adaptive immune system. So the types of cells that we see in the gastrointestinal mucosal surface are numerous. There are at least 7 different types of cells, and I'm just going to give you a little bit of information about some of the ones that I deem to be very important. Let's start with a cell type called the goblet cell. The goblet cells are found throughout the intestinal epithelial surface, and these are specialized cells that have secretory vesicles that contain mucin. And these goblet cells secrete this mucin, and at the very left of this slide, at the top, you see the physical barrier that mucin produces. This also provides an area for binding sites that inhibit certain antigens from getting through to the epithelial surface. And it's also a link to the immune system. Immunoglobulins, IgA, for example, are sometimes found in this particular barrier. We have cells in the subepithelium, such as plasma cells, that produce IgA, IgG, IgM. And these are very important in terms of containment. They actually act as barriers to certain microbial components getting through the gastrointestinal tract. We have Paneth cells. Paneth cells are found at the very bottom of the crypt villus axis. And they are very close to the region of the intestine where you see a lot of mitosis occurring. There is no mitosis in the villus. It is only in the deep crypt region where you see mitosis. And these Paneth cells secrete defensive molecules that protect the mitotic region of the gastrointestinal tract. If you lose that mitotic region, the intestine dies. So these are very important, and the defensins produced by the Paneth cells are thought to be very important. Another cell type is the microfold, or M cells. And these are located in the intestine that overlie lymphoid tissue. And M cells allow for translocation of certain antigenic substances. And they're highly selective in allowing for this translocation of these antigenic substances. These are cells that require a lot more study. They're very fascinating, and I think that we know very little about how they actually work and their actual function. We also have the intestinal epithelial cells, the regular intestinal epithelial cells, that have different junctional complexes. And the tight junction at the very apex between intestinal epithelial cells is thought to be very important. So if you think of my hand and these fingers as being the cells of the epithelial cells of the intestine, if these junctions break down, the epithelial cells spread apart. And as they spread apart, what can happen? You can have translocation or increased permeability of the gastrointestinal tract. So anything that causes loss of those proteins in the tight junctions of epithelial cells can lead to intestinal dysfunction. And here are just some of the disease entities that have been associated with a leaky gut, hyperpermeable intestine, sepsis, systemic inflammatory response syndrome, inflammatory bowel diseases, celiac disease, necrotizing enterocolitis in babies, type 1 diabetes, atopic diseases, and even autism have been associated. None of these have been proven causal, but there is an association between increased permeability and some of these disease entities. Now let's go back in history a little bit. And this lady that you see on this slide, Christiane Nusslein-Volhard, won the Nobel Prize. And she was working in Tübingen, Germany. And in Tübingen, one day, several decades ago, one of her graduate students was working on Drosophila. And Drosophila, as you know, is a very commonly used model for looking at developmental biology. And they were looking at Drosophila that had certain mutations. For example, where the antennas were coming out where the feet were supposed to be coming out, vice versa. So this graduate student did protein gels on these different Drosophila and found some differences in the protein gel. And the graduate student went to Dr. Christiane Nusslein-Volhart and said, and showed her the protein gel, and Dr. Volhart said, "Das ist ja toll." Okay, some of you who know German will know this is cool. Toll means cool, okay, or really great. Well, these proteins then got the name toll proteins. A few years later, this gentleman, Charles Janeway at Yale University, was doing some work on various intestinal cell types and also macrophages, and he found that these macrophages contained toll proteins, and that these were very important for transduction of signaling, microbial signaling, viral signaling, bacterial signaling, fungal signaling to these to the cells and also to the rest of the immune response. A few years later, these three gentlemen also won the Nobel Prize, and two of them won a quarter of the Nobel Prize, and the third one, Ralph Steinman, seen furthest to the right, got 50% of the Nobel Prize. Why did this happen? Well, Ralph Steinman is the guy who was doing more than just looking at the innate immune system. He was also looking at cells that were of the macrophage cell type, and these were dendritic cells, and found that this particular cell type was the link from the adaptive immune system to the, from the innate immune system to the adaptive immune system. So as an example, these dendritic cells in the intestinal mucosa can link to the luminal surface and sense what kind of antigens are in that luminal surface. They transduct that signal. And then in the submucosa, they interact with effector T cells and T cells that are tolerizing T cells and help them differentiate into either tolerizing T cells or effector T cells. So they start out as non-differentiated, and with the interaction with the dendritic cell, they turn into effector T cells or tolerizing T cells. Also a very important component of our immune system. Now, what about these toll-like receptors? And we now know that there are several toll-like receptors. There's at least 10 of them. And here's some examples of some of these toll-like receptors. The ones that appear to play a large role, at least in my field, are TLR4, TLR2. And these relate to various bacterial components. Work that was done at Yale University several years ago by the group that was started by Dr. Janeway. The chief of that group was Dr. Ruslan Medjetov, published this paper in Cell. Recognition of commensal microflora by toll-like receptors is required for intestinal homeostasis. So they were asking the question, are these toll-like receptors really important for maintaining the gut integrity? And so what they found, and I'm not going to go into the detail of this, but they found that low-grade stimulation was very important, or sort of like tickling of these toll-like receptors by commensal bacteria was very important in terms of maintaining gut integrity and maintaining gut homeostasis. I can't get into all the details of their study, but it involved knockout animals, TLR2, TLR4 knockout animals, putting microbes back into the solutions that these animals were eating, and also treating them with very broad spectrum antibiotics. But I don't have the time to get into all that, but you could look at that article. Very interesting article. I think it's a classic. So commensal microbes have many beneficial effects for the host: nutrient metabolism, tissue development, resistance to colonization with pathogens, and maintenance of intestinal homeostasis. Now, what about the microbiome? As a result of the Human Genome Project, which was actually started in the late 1980s, early 1990s, new technologies developed. And these high throughput technologies were applied to the microbes. And if you look at culture-based technologies, it's very difficult to use all of these different culture-based technologies to be able to find all the different microbes in a certain niche, for example, in our gastrointestinal tract. But using these newly developed techniques, 16S, metagenomics, et cetera, whole genome sequencing, we find that we can actually identify many, many more taxa of microbes than we were ever able to determine in the past. We have regional differences in the gastrointestinal tract. And in the very upper part of the gastrointestinal tract, in the duodenum, there are very few microbes. As you go further in the gastrointestinal tract into the ileum, you see about 10 to the 6th microbes per gram of fecal material, and you go into the colon, you see about 10 to the 12th, okay? So many more. You have this aboral gradient of microbes, and they play different roles in different parts of the gastrointestinal tract. So in the distal intestine, distal small intestine, they play more of an immunologic role, whereas in the colon, they play more of a bioreactor role, where they ferment certain foods, and these fermented foods, for example, if you ferment foods into short-chain fatty acids, acetate, propionate, and butyrate, this can be very important for various aspects of the gastrointestinal tract. So, for example, butyrate, if you have a cell culture system with CACO2 cells, for example, and you do not put butyrate into the CACO2 cells, it takes several days for those cells to become confluent. But if you put butyrate into the system, those cells become confluent much more rapidly. So they actually help in developing the junctional integrity of the gastrointestinal tract. Now, when we get to my field, neonatology, human milk seems to be very, very important for helping our babies, our preterm babies as well as term babies. And in terms of the microbiome, many studies have now been done on the microbes that we find in human milk, and they are certainly present. In the past, in the neonatal intensive care units prior to several years ago, we would occasionally culture some of these milk samples and find that there were bacteria there. And when we saw bacteria that we were able to culture, we threw it away. So, yuck, can't give that to our babies. But as time went on, we were beginning to recognize that these microbes may play an important role. This is a study that was done at the University of Idaho that looked at 16 different milk sample, or 16 different mothers providing milk at 1 week, 2 weeks, and 4 weeks after birth, okay? And the colors here represent different phyla of bacteria. So this is one mother, this is another mother. This is at 1, 2, and 4 weeks. And within that one mother's milk samples, at 1, 2, and 4 weeks, you can see that the colors are very similar. But then you go to the next mom, you again see the colors seem to be very similar. They change a little bit during time, but they're very different from one another, suggesting a personalization of the microbes in that milk from each mother. Very interesting phenomenon. Now, we've been touting the use of probiotics for quite a while. And it's a very long story that I'm not going to have the chance to get into. But if you think about the microbes in human milk, and if you assume an intake of 800 milliliters per day, this is a healthy term baby. And if you assume 10 to the 5th to 10 to the 6th bacterial cells per milliliter, This will provide 10 to the 7th to 10 to the 8th bacterial cells, which may be personalized daily, close to the dose in most of the probiotic studies that have been done in our babies. There's also this dynamic interaction, something called the enteromammary immune system, that was recognized even back in the late 1970s. This is a paper from Drs. Kleiman and Walker. And the way this works is that if there is close contact between the baby and the mother, that baby may not have a mature immune system, but the mother has a mature immune system. And if the baby gets colonized by certain microbes that might be pathogenic, that baby may not be able to respond very well to those microbes, but the mother can. So with a close contact between the mother and the baby, these microbes get into the mother's mouth, into her gastrointestinal tract, and her immune system produces the antibodies to those particular microbes. And they get secreted into the breast milk. And over time, we've begun to recognize that this actually does work in humans. We've seen several studies now showing that the existence of an intramammary system. I can't get into the details of those studies because of time limitations. So, of interest is that we've done a lot of work now with the microbiome. And one of the areas that we've been working with at the University of Florida is necrotizing enterocolitis. And we have found that the microbiome in babies who develop necrotizing enterocolitis is very different than those babies who do not develop this particular disease. And it's been thought that perhaps administering probiotics might help. Most of these studies of the microbiome or studies of metabolomics, et cetera, if you look at only one of the omics, this is potentially problematic, especially when it comes to an association with subsequent development of a disease. Because this is only an association. It does not prove causality. So we're getting into a different era now where we are using something called multi-omics and integrated multi-omics. So in the middle of this picture, we see several layers of omics. We see the genome, epigenome, transcriptome, proteome, metabolome, et cetera. And we need to be able to integrate this to be able to determine causality. And an example I really like to give is shown on the very left slide, part of this slide. You see this picture of an elephant. Well, this is the parable of the 6 blind men of Hindustan. Each one of these blind men was asked to feel this structure. And one went to the side of the structure and said, "This feels like a wall." One went to the back of the structure and said, "This feels like a rope." The other went to the front of the structure and said, "This feels like a spear." They got together, they argued, they were adamant in what they felt. But they didn't really integrate their thoughts. If they would have integrated their thoughts, they would have figured out that this was an elephant. The same thing holds true for multi-omics. We are developing some of the technologies now to integrate multi-omics. And so, in the future, using another new technology, artificial intelligence, we will be able to better cluster different phenotypes of individuals. And with this clustering, within each single cluster, we should be able to do the integrated multi-omics to determine a mechanism of what is actually going on within each one of those clusters. And then use a systems biology approach to develop biomarkers that will tell us early on, before any kind of a problem develops, which individuals will be at highest risk for development of certain problems. And then with these biomarkers, we can then intervene. Again, this is very early We're just putting our foot in the water with some of these studies, but I think that this is a direction we are going in the future. So I'd like to stop here with some take-home messages. Perturbations in the early mucosal immune system may have lifelong consequences. And this is one of the reasons I really enjoy working in the field that I'm in, because sometimes the things that we do to our babies have lifetime consequences. But we have to be really careful because we can mess up, and that can have lifetime negative consequences as well. Microbes and early nutrients likely play an important role in mucosal immunity, both before and after birth. And newly developed tools such as artificial intelligence and multi-omic analysis will aid the understanding of the effects of newly developed agents on the developing individuals. And I'm going to stop there. Thank you for your attention.